Good morning. It's really a pleasure to be here with you today, and I appreciate Dr. Gardner's talk introducing CAR T cells. And really, my goal this morning is to talk about expanding the use of CAR T cells and applying this to the treatment of brain tumors really as a model for what needs to be done to advance this therapy for the treatment of solid tumors. And so I have the following disclosures. So, as we know, what's really changed the landscape and what was elegantly presented in the previous talk of CAR T cell therapy is the remarkable clinical success with CD19-CAR T cells. And so I'm just highlighting one example of a patient treated at our institution, City of Hope. This is an individual that had relapsed high-grade B-cell lymphoma, and after lymphodepleting condition and a single infusion of 200 million CAR T cells, you see complete tumor eradication. And so this has really changed what we believe these cells can do, and it's really due to optimizations at all steps of the therapy. This includes, over the last several years, optimizations in CAR design, T-cell manufacturing, and aspects of adoptive transfer that all integrate to improve this engineered therapeutic cell for optimal recursive killing, cytokine production, and the ability to proliferate on antigen. And this is really what allows or has mediated these remarkable clinical successes in B-cell malignancies. So now really where the field stands is, can we extend this therapy beyond CD19? And what are the challenges for solid tumors? So if we go through some of these challenges, they include trafficking, the ability to recognize solid tumors and kill, proliferate within the tumor microenvironment, and counteract the suppressive effects of the tumor microenvironment. And so these really lead to many questions. Will more advanced CAR designs be necessary to target solid tumors? And what antigens can be safely and effectively targeted? And how can solid tumor heterogeneity be overcome? And also, how does the suppressive microenvironment limit the therapy? And also aspects about dosing and how do you deliver these T cells. So these are really some of the questions I want to address today as our group at City of Hope has been applying this therapy to glioblastoma. So glioblastoma is the most common primary brain tumor and one of the least curable of all solid tumors. These are highly aggressive tumors that can grow rapidly, as shown here in this patient in just 4 months, how fast these tumors grow. And even with aggressive standard of care, 5 to 10% of the patients will only survive less than 5 years after initial diagnosis. And as we look at median survival, improvements in median survival have been quite modest over several decades. So this has really inspired the idea of using immune-based therapies to improve outcomes for patients with glioblastoma. And I think possibly an underappreciated fact, but maybe came out of the last presentation, is that really some of the lessons learned from CD19 CAR therapy has really inspired and energized the field to apply CAR T cells to brain tumors. This includes the observation, as presented previously, that CD19 CAR T cells can traffic to the CSF and can reduce incidence of leukemia in the CSF. But also, and so I think most CAR centers today, because of these observations, either have preclinical or clinical programs trying to optimize this therapy for the treatment of brain tumors. I think what's also given a bit of pause is the neurotoxicity that can be associated with CD19 CARs, and really highlights potentially some of the challenges that may be associated with applying this technology to brain tumors that we'll discuss. So when we think of the challenges, what I want to present today is looking at target antigen selection, tumor heterogeneity, microenvironment trafficking, and some early clinical applications. These are challenges that exist for all solid tumors, but for my presentation today, I'll be mostly focused on its application to brain tumors. And so first, target antigen selection. You know, it's clear that we can engineer CAR T cells to recognize tumors and solid tumors. So that's not really the question. The question is, what can be safely and effectively targeted? So when thinking about applying this technology to brain tumors, you really cannot have off-tumor targeting of normal brain. So the barrier is much higher. So really the ideal antigen cannot be expressed on normal tissue. Ideally, it would be broadly and uniformly expressed on malignant cells and have a role for promoting tumor growth. So thinking about the handful of tumor antigens currently being evaluated for targeting glioblastoma, really one of the lead targets is the tyrosine receptor kinase EGFR, and that's due to its amplified in the vast majority of GBMs, but really what's been limiting its utility for CAR therapy is the known expression on normal brain. Therefore, most institutions focused on EGFR are targeting the most common mutated form of EGFR, EGFR-V3, and this is being pursued by multiple groups, both preclinically and clinically. HER2 is also a very attractive target for GBM as it's expressed on the majority of primary brain tumors. Although compared to expression in many breast cancers, it is not amplified. Our group has been focused on targeting IL13 receptor alpha 2, a high affinity IL13 receptor that was first shown by Dubinsky and colleagues to show the strict expression on malignant tissue versus normal brain. And then there's other platform or targets being evaluated for GBM, but aren't quite as developed. So focusing on sort of the three lead candidates I'd like to talk about today, EGFR, HER2, IL13 receptor alpha 2, what we see is that these are broadly expressed in GBM as shown here and often co-expressed on the same tumors. I think what's concerning is that these targets are often expressed in many normal tissues. So it really creates this challenge of how do we make sure we can target these antigens with CAR therapy and minimize toxicity. And so it really comes down to fine-tuning CAR design. CARs are synthetic immunoreceptors that are modular with the tumor targeting domain that determines the overall affinity, the extracellular spacer domain, and the intracellular signaling domain. And it's really the integration of all these structural components that determine the overall specificity of the CAR. And so just to highlight a little bit, a few studies really focused in brain tumors targeting EGFR to improve selectivity. It is clear by modulating the affinity of the tumor targeting domain that we can improve selectivity either for the mutated form of EGFR-V3 or amplified EGFR over endogenous EGFR. So this may be as a strategy, overall strategy, by fine-tuning the affinity of the targeting domain to give better selective targeting of amplified receptors, cell surface receptors. Other approaches being developed include masking chimeric antigen receptors, so taking a masking peptide that blocks the CAR from targeting, in this case, EGFR on normal tissue, but it's linked with a protease-sensitive linker. Proteases being upregulated in the tumor microenvironment, the model is that this would be cleaved specifically in the tumor microenvironment, giving selective activation. So these are just a few of the examples where the field is trying to improve the specificity of these CARs to broaden the repertoire of antigens that we can use for solid tumor targeting. And so on the flip side, though, other than improving specificity, we have to be really cognizant of the aspect of heterogeneity. We see this in CD19 CARs, where antigen escape variants is one etiology of disease relapse, and this is particularly important for solid tumors. So GBMs are highly heterogeneous tumors. There's inter-patient or patient-to-patient heterogeneity as defined by the TCGA of different molecular subtypes. And then within tumors, there's great heterogeneity, and even at the cellular level. So what this allows are cell populations that exist within the tumor that can invade monotherapies and then lead to disease recurrence. And so there's a lot of effort really focused on developing multi-targeted CARs. These could be mixed CARs, so it could, in a precision medicine way, where you would evaluate a tumor and then engineer to recognize with different pools of CARs that recognize the tumor of interest. Or they could be a single cell product, either with expressing two independent CARs or a CAR that has dual specificity. And these are all in early stage preclinical development. And so in our group, we've been focused on these mixed CARs and can show by mixing CARs that have specificity for receptor alpha 2, HER2, and EGFR, we can actually inhibit antigen escape in preclinical models of GBM. The Baylor group has put a lot of effort and have elegant studies really targeting HER2 and IL13 in a tandem CAR design. And their work elegantly shows that the tandem CARs outperform either dual and mixed CARs in their models. So I think there's a lot more studies that need to be done, but this idea of expanding or limiting antigen escape through multi-targeted CARs is very attractive. But another really interesting observation came from preclinical models by the Sampson lab at Duke in syngeneic mouse models. And what they showed is when they targeted an EGFR-V3 tumor with EGFR CARs, they actually got tumor elimination, but these mice were then protected with re-challenge of tumors that were EGFR negative. And so this data suggests that the CAR T cells might stimulate a host immunity against other antigen targets. So this is also a very attractive idea that CAR T cells themselves, by even targeting a single antigen, might promote antigen spread. So whether it's mixing CARs or promoting an endogenous immune response, this idea of overcoming tumor heterogeneity we know will become a major issue for solid tumor targeting. So now I'd like to turn briefly to the microenvironment. I think this could be a talk in itself. We know the microenvironment is highly suppressive to the function of T cells, their proliferation, their persistence, their cytolytic effector function. And this is through a variety of mechanisms: the presence of immunosuppressive immune subsets, immunosuppressive cytokines, inhibitory receptors, nutrient starvations, and even the architecture of the tumor itself. And so there's been many strategies that are currently under development for both brain tumors and solid tumors in general to counteract these suppressive effects of the microenvironment. And just to kind of highlight a few, and this is definitely not an exhaustive summary, there's a lot of interest in combining CAR T cells with checkpoint blockade. So it's clear that checkpoint blockade, or it's clear that PD-1 upregulation can promote, or CAR T cells are susceptible to PD-1 exhaustion. And so there's strategies that are being evaluated to combine CAR T cells with either anti-PD-1 antibodies or dominant negative approaches or ways of knocking out PD-1 in T cells through CRISPR or shRNA. And then there's also strategies of actually transitioning these inhibitory receptors to be more co-stimulatory, and this is being used both in the PD-1 arena as well in suppressive cytokines. So the same approaches, whether we make dominant and negative receptors that inhibit the signaling of these suppressive cytokines or turn them into a co-stimulatory signal for the CAR T cells. And finally, there's approaches that are being used to really remodel the tumor microenvironment, and the idea of using CARs as delivery pharmacies, and exciting work by several groups really looking at the delivery of IL-12, either constitutively and induced, to really promote a more anti-tumor microenvironment. And so I'd last like to talk about really the challenge of tumor trafficking for solid tumors. Here's a video. We'll see if it plays. I had some problems earlier, but these are CD8 T cells that are engineered to express the firefly luciferase. And what we see with a glioma tumor engrafted in the flank, we can see that these cells can efficiently traffic to the tumor. And this is really a prerequisite for efficient solid tumor therapy. But the question becomes, in a lot of these solid tumors, and particularly brain tumors, what is the optimal route of delivery? So it's clear in the CD19 field that T cells can traffic to the CSF and mediate efficacy. But in primary brain tumors, the question is this the most effective route of delivery? And so we've been interested in actually comparing systemic intravenous delivery with more local, regional, or intracranial delivery. So thinking about IV delivery, really, this is probably more clinically friendly to deliver these cells IV, but then it does require these cells to be able to traffic and cross the blood-brain barrier, and there is likelihood that it would increase the risk of off-tumor toxicity to normal tissue in the periphery. But as I mentioned, there's substantial evidence that the T cells delivered IV, whether it's with tumor-infiltrating lymphocytes that are melanoma-specific, CD19-CARs, have bioactivity. The other alternative approach would be to deliver these cells locally, and this is attractive because this is really a local disease, right? And so this could be achieved by delivering these cells at the site of the tumor or into the CSF so that they could potentially better traffic to all sites of disease, infiltrative and multifocal. This approach then requires a device, a reservoir catheter device, but on the positive side, and something that we've found very useful, having this device locally actually allows for local sampling, so you can actually monitor what's the microenvironment at the site the T cells are delivered and where you want their activity to be monitored. And so our group has provided clinical evidence in primary brain tumors that local delivery also provides bioactive T cells, and I'll present some of this work at the end of the talk. So we've spent a lot of, really focused preclinically on trying to understand in mouse models different aspects of delivery. And so we've compared in our primary GBM models the ability of intratumoral delivered T cells compared to IV delivered T cells and their therapeutic benefits. So in these NSG mice models of human GBM, we deliver T cells IV or ICT into the tumor bed, and what we see in these models, it's really only the T cells delivered locally that give long-term survival. And in fact, we were quite surprised that IV delivered T cells in this GBM model really provided no therapeutic benefit. when we went back and looked at the tumors, we actually found that the T cells did not traffic well to these GBM tumors. And so we've gone on to test this in multiple tumor models. We've recently looked using our HER2 CAR T cells and compared intraventricular delivery, where we deliver the cells to the CSF, compared to IV delivery. And in this model, again, if we give single infusion of 500,000 CAR-modified cells, gives long-term survival in all mice treated, where IV-administered cells, 10-fold greater dosing gives survival benefit in about 50% of the mice. And so what we in multiple models see, it's not that IV-administered cells don't provide therapeutic efficacy, but on a per-cell dose level, really we get more effective therapy at lower doses with local delivery. And so this has really provided the rationale for evaluating this clinically. And this is not unique. There's other studies coming, that have been published in other solid tumor models where intratumoral delivery seems to outperform IV delivery. And so we'll see if this is a common theme. And so, why is this? What is the difference between this and what we're seeing in CD19 T cells? It's well established in models of autoimmunity as well as CD19 that activated T cells are more efficient at trafficking across the blood-brain barrier, so this might be one aspect. And also IV therapy may be improved by preconditioning, and there's preclinical data to support that. But really, my belief is that we're going to understand more about delivery by actual clinical evaluation. And so looking at the clinical trials that are ongoing, really there's groups testing these cells by IV delivery, and our group, as well as Baylor, is looking at a local approach for now. And so what I'd like to do in my remaining minutes is focus on early clinical results, really putting all these themes together. And so, if we look at the trials, the CAR T cell trials worldwide being applied to primary brain tumors, there's really 12 trials, 5 in the U.S., and really focusing here in the U.S. only because of time. 3 of these trials are focused on targeting EGFR-V3. This is a trial out of UPenn, the NCI, and Duke. And so what I'll talk about today is some early clinical findings, work that has just been published in the last few months from our group using CAR T cells to target IL13 receptor alpha 2, and the Baylor group looking at HER2 specific CAR T cells. So the Baylor group just recently published a very elegant study evaluating HER2 CAR T cells and their platform really focuses on engineering viral-specific T cells. These viral-specific T cells are expanded from peripheral blood and show reactivity to CMV, EBV, and adenovirus. These viral-specific T cells were engineered with a second-generation CAR, and this is an attractive approach because it's been reported that many GBMs upregulate CMV, and so it's attractive to think that maybe the CMV specificity might give the CAR product 2 ways of recognizing the tumor. And so this is really a dose escalation study, and patients receive 1 to 4 IV infusions. They reported the treatment of 17 patients, treating both pediatric and adult patients that have had recurrent disease, so had undergone standard of care and had several surgeries, temozolomide, and often several salvage therapies. And so what I think is exciting is that it's really the report that we can target HER2 for GBM in a way that's safe. There were no reported DLTs. And really, 8 of the 17 patients showed some clinical benefit. And here I'm showing their report of a partial response of a patient with a non-resectable GBM. And you can see how the decrease in tumor burden here over time after CAR T-cell infusion. So this is really exciting for the solid tumor field. The idea in such a challenging disease setting, we can see a partial response. And the Baylor group went on to look at CAR T-cell persistence and proliferation, and what they report is that these CAR T cells were detectable in the blood for up to 12 months, but they did not observe any systemic T cell expansion. And it really raises the question, what compartment should we be analyzing when we're looking at responses in the brain for T cell expansion? Because the blood has been very informative for B cell malignancies to follow expansion and persistence. And so their group also reported that 5 of 17 patients showed edema at the glioma site with their IV administered cells, really suggesting maybe changes in the local immune microenvironment and possibly suggesting trafficking of the therapeutic cells. And so our group has really been focused on targeting GBM using CARs engineered to recognize IL13 receptor alpha 2. The therapeutic platform is a second-generation CAR with a 4-1BB co-stimulatory domain. The CAR cassette, again, contains a truncated CD19, similar to the truncated EGFR-T, so it's a way to follow the gene-modified cells. And it's an engineering platform where we actually pre-enrich cells with a central memory phenotype, and then lenti-engineer these less differentiated memory cells. In our preclinical models in mice, single infusion of these CAR-modified cells show potent anti-tumor activity, and as few as 100,000 cells give survival benefit in the majority of the mice. And so this really was the rationale for initiating a phase 1 trial. We treated our first patient almost 2 years ago. And so the key features of this trial were enrolling patients where we confirm expression of the target antigen, receptor alpha 2. We're actually looking at multiple local delivery routes. And so patients are enrolled during CAR manufacturing. They undergo surgical resection and placement of this reservoir catheter. And then individuals are given weekly infusions of CAR T cells. And so just to summarize some of our interim results, so we've now treated, as of March, 10 patients on our resection arm, where patients undergo surgical resection of tumor and then T-cell administration. 8 of 10 patients show clinical benefit with stable disease or partial response, and many of the patients are alive out 6 months. This is actually really encouraging because many of these patients are, or all of these patients are heavily pretreated, and so we're encouraged by these early findings. Also important is that, Also important is that targeting IL-13 receptor alpha 2 has been very well tolerated. We saw no DLTs similar to the HER2 study by Baylor, no cytokine release-like syndrome events, and no neurotoxicity. And so now I'd like to just finish with one patient that has been very informative to our study that we really that we just published in New England Journal of Medicine, where this stable disease was then resulted in a CR after additional T-cell infusions. So this was an individual with recurrent multifocal GBM. He had 5 tumors in the brain, very poor prognosis. When he came to us, his oncologist said he had less than 2 months to live. And so we actually are the neurosurgeon at City of Hope, my collaborator Benim Badi, resected 3 of his 5 intracranial tumors, and his largest tumor here placed the reservoir catheter system where 6 infusions of CAR T cells were administered. So we're encouraged by the fact that over a 45-day period, we saw no tumor recurrence at the site of injection. However, his other tumors, or near the other sites of resection, he was actually recurring and progressing. So these tumors were getting larger, and he actually had a large lesion in the spine. And so based on some of the preclinical work that we had done looking at intraventricular delivery, we actually went to the FDA on a compassionate use protocol to ask for the ability to infuse these cells into the CSF, and this would be the first patient in which CAR T cells are actually delivered directly to the CSF. And what we found was quite remarkable. After just 3 infusions, we started to see dramatic anti-tumor response, and after 10, his tumors in the brain became really undetectable by MRI. And very encouraging, we saw complete elimination of his spinal tumors. So this was a disease setting where actively growing tumors were progressing, and by delivery of CAR T cells, we're able to achieve a complete response. Graphing all lesions, we see that all lesions responded to T cell therapy. And similar to the local intratumoral delivery, the delivery of CAR T cells to the CSF was also safe with no CRS or neurotoxicity. And what was very encouraging is that he was really able to return to normal work and life activities. So while that's encouraging, unfortunately, the durability of the response was only 7.5 months after the initiation of therapy, and he eventually recurred. And so this really leads us to understand, why did this recurrence happen, and what can we learn from the initial challenges I laid out at the beginning. And I think the most important is that his recurrent tumor actually showed lower expression of our target antigen. And so this really highlights the importance of antigen escape and the heterogeneous nature of these solid tumors and how we might address this. But it was actually very interesting because it was more than just loss of IL-13 receptor alpha 2. His tumor completely changed upon recurrence. So initially his tumor was very high HER2, low EGFR. His recurrence then was low HER2, high EGFR. So I think it really highlights maybe a more complex phenomenon where there's really complete immune editing of the tumor. And finally, another lesson learned from this study is that each intraventricular infusion of CAR T cells here is represented by a dotted line, and what we saw is that there was an influx of T cells within the CSF after each infusion. We thought these were likely CAR T cells because this is what we were delivering, but to our surprise, very few of these cells were CAR modified. The majority were endogenous CD3 cells and mature myeloid population, CD14 HLA-DR. And so this is very interesting to us. How does the infusion of CAR T cells then influence endogenous T cell responses? And this is really an active area of investigation for our group. When we look at T cell persistence, we can actually see these T cells persisting over the weekly infusion cycles as shown here, but we're also, similar to what was seen in the HER2 study, not seeing dramatic, we're not seeing CAR T cell expansion locally. So just to summarize now, with taking a step back, I think looking at these early clinical trials, I think is encouraging. I think, really the first hurdle when we start to apply CAR T cells to solid tumors is to understand what can be safely targeted. And so I think we're making good progress there. So thus far, there's reports by our group, Baylor, and others that IL-13 receptor alpha 2, HER2, and EGFR-V3 are antigens that can be safely targeted for GBM. And these unique responders really kind of highlight the therapeutic potential. Not all patients are responding in this way, but it says it is possible, and we have to understand ways of further empowering the therapy. And so that really gets to expanding the repertoire of targets, boxing in tumor antigen escape, and establishing what the optimal delivery and dosing is. And then likely for most patients, it's not going to be just a CAR-engineered T cells. There's going to have to be more complex engineering approaches to overcome the suppressive microenvironment. And ultimately, our group and others really want to expand CAR therapy to many other brain tumors. We are now, we'd like to apply this technology to pediatric brain tumors, and in the next few months we'll be opening up a trial targeting HER2 for breast amplified HER2 tumors that have metastasized to the brain. So we're very excited about that. And so with that, I'd like to thank our team at City of Hope and answer any questions. Yes. So the question is whether we've considered testing lymphodepletion, and I think that's a really interesting question. We have not incorporated lymphodepletion in this clinical trial, really because of the timing of surgery and the delivery of the cells, and we weren't sure whether local delivery required lymphodepletion. But I think it's a very interesting question because you could really change that microenvironment possibly, and maybe the homeostatic cytokines would improve. So it's something we definitely discuss, and I think it's a very interesting question. Thank you.
Is the myeloid infiltrate in the CNS, is that good, bad, indifferent, and do you want to block it or not block it?
Right, so what I can tell you is that it varies from patient to patient. And we think, so the question is about the myeloid infiltrates. And for this patient that had the response, they were mostly CD14 and mature expressing HLA-DR. For other individuals, they're CD15, arginase positive, immature, and look very suppressive. And so what we're trying to understand is which subsets are infiltrating and how that might respond to therapy. So we actually think the different subsets will be impacting the therapeutic.
I was wondering if you had any additional information on the differences in the great responder versus the other patients in the trial. Any more histology differences that you might think are contributing to why this patient responded for 7.5 months and others ones didn't do quite as well?
Right. So one thing that we're looking at is sort of the baseline tumor microenvironment at the time of treatment. And the individual that we treated that responded quite remarkably, he did at the time of treatment have quite a few immune infiltrates. And so at the time of treatment, so you know, in this idea of hot versus cold tumors in the spectrum, so GBMs are thought to be more cold tumors in general, but in that spectrum, his tumor might have been a little more hot than other tumors. And so we're actively investigating that and to try to understand how the interplay between CAR T cells and the endogenous immune system might impact overall outcomes.
Thank you.
Thank you very much.
Okay, for our last speaker, we'd like to introduce James Riley from the University of Pennsylvania, and he's going to talk to us today about CAR T cells approaches as part of the HIV Cure strategy.
Good morning. And as Matty Python said, now for something completely different. There's my disclosures. A question that's been lingering in the field for quite some time is, can HIV be cured? And because at this current point in time, we have very good medications, but it is sort of an open question. Can we sort of in a reasonable success rate cure people with HIV? And at this point in time, there's one person who will definitively say yes. And this person, Timothy Ray Brown, has quite of an interesting pathway that's diagrammed on the slide there, which he was diagnosed with HIV, gets leukemia, almost died, he returns, and then was treated with a bone marrow transplant from somebody who had a mutation in their CCR5 gene, which CCR5 is a critical receptor for HIV entry. And since around 2007, he has been completely HIV negative. But this was repeated. The problem in these types of things, the patients have a very different clinical history, so it's very hard to perfectly match a protocol. But similar ideas were tried, and Timothy Ray Brown remains the only person who actually was cured. Most of the patients actually died of leukemia because of it came back too quick. And so while it's a great proof of principle that HIV can be cured, it's certainly not going to be the way we cure HIV in everybody. And so why can't we cure HIV with the current drugs we have? And so here lists a couple reasons. It's unclear whether the drugs actually fully suppress all HIV replication. And just a little bit of replication radically changes the math involved to get rid of all the HIV. But the main culprit in this are what we call latently infected T cells. And the word latency is probably not the greatest word because in HIV it's largely just a mistake. The cell largely integrates in a cell that was about to rest down, we think, and there just isn't enough metabolism to sustain it. And so here the virus is immunologically silent, and at some frequency it will reignite and reseed the population. So this is why people need to stay on their medications for their entire life. Didn't mean to do that. Oh crap. Sorry about that. So the current thought in the field is how can we activate this reservoir and then eventually purge it? And it's been called the shock and kill approach, in which a number of agents are supposed to tickle a T cell. And this tickling is a very complicated process because you want to give it enough of a signal so that HIV gets reactivated, but it can't be too much because then you'll have massive cytokine storm. And so there's a sort of a Goldilocks attempt is to get enough things to get it going. And under no one's belief that this is going to work 100% of the time, every time. And so you can imagine that there'll be multiple attempts to tickle the reservoir. And while some argue that it may be possible to fully eradicate the virus, I think that's asking quite a bit. But could you lower the amount of virus and that have a clinical benefit? I think that is definitely possible. And then the second part is the kill part. And so the thought is if you induce the HIV, the immune system will then now recognize the infected cell and then kill it. And unfortunately, that's where the problem lies right now. So there are a number of agents with varying efficiencies that people have shown can induce a viral spike when administered to people. But that spike is not sufficient to actually kill the cell. And Bob Silicano's group showed that you actually need CTLs to do the killing, but then he also showed that most of the virus that comes out has escaped from the patient's CTLs. And to make things even more complicated, the drugs that we administer, many of them are not very healthy for activated T cells. And so the shock is actually killing your killers, which probably isn't the best way to go forward with this. And so what I'm going to try to argue with you today is why we need bionic T cells. And this is a great crowd because you're all here because you believe. But the rest of the world might need some convincing on this. And the argument goes is that HIV has done a pretty good job of escaping the natural immune response. And at the time, so while ART is great, the one thing it does do is it lowers HIV antigen. And so when that happens, your resident T cell response goes down, and the response that remains is largely defective. So at the time when you want the T cells to be at their best, they're weak and diminished. And so we would propose that, since we know exactly when we're going to do this, we can infuse genetically altered T cells, presumably very high doses, and those cells would, one, be functional, and they could be engineered so they could arguably be better than your endogenous T cell response. So for those of you, this looks like a reasonably mature crowd. So many of you actually might have seen The $6 Million Man, which was one of my favorite growing up. And so in the thing, he's an acid he gets burnt up, and and the key point of this is that they just didn't want to fix him. They wanted to make him better than he was. And I think the same argument could be used for the HIV specific T cell, because it crashed and burned the first time HIV got in there. And if we're going to rebuild it, we need to make it better than it was before. And what I'll try to go over today is that we need several key points of this. The first thing to do is we need to protect the cells from getting infected. Otherwise, they're going to crash and burn again. And then through the use of CAR T cells, we can sort of change the rules by which the T cell response engages HIV. And hopefully HIV hasn't figured out how to escape from the CAR approach as well as is effective from the natural response. And something that is the great question in this is, how can we sustain these T cells? Because if we do a good job, we should eliminate most of the antigen. And we know that the T cell levels are highly influenced by the amount of antigen present. And so this is, if we're thinking about curing people for 10, 20, 30 years, we just don't really have much of a working knowledge base on how we can sustain these cells to live. So I just really want to go quickly over a trial that we did several years ago, because this gives me great hope that maybe we're not all that far away. And so this was a trial, and I'll run through it really quickly because of time, but CCR5 was targeted through zinc fingers, and these were infused into patients, and then the patients were taken off their therapy, and the time to rebound was calculated. And then if you sort of do the back of the envelope math, roughly 10 billion cells were infused, but of those 10 billion, only about, and this is probably a generous, but it makes the math easier, only about 10% of those were actually CCR5 defective. So we're putting 1 billion CD4 cells that are resistant to HIV in a background of 500 billion CD4 T cells. So we're not stopping the spread of the virus. There's plenty of places for HIV to go. But what we saw is that in most patients, well, in all the patients, when you take them off their therapy, the virus comes up. But in most of the patients, we saw roughly a log and a half decrease once the virus came up. And then there was this one patient, this green one right here, and he went down to limited detection before that. Now, in this particular data, we had to start him back on ART again, so we didn't know. But what we've done subsequently is treated several other patients, and the patients who respond the best to this therapy are ones that are Delta 32. So they have one defective CCR5 gene to start with, and so when you do the CCR5 disruption, it gives you more defective resistant viruses. And we've seen some remarkable times. So Segmo did 3 patients in which some of them were off medication for 50 weeks. We've just repeated this. Actually, with this patient, we reinfused him again, and this time we were able to keep him off medication, and he stayed off drugs for 35 weeks. And so, is that a cure? No. But if you consider how few cells we actually put in, and now we have better ways of engineering the cells, I'm reasonably optimistic that we can get control of the virus. So there's a long and great history of collaboration between HIV and cancer immunotherapy. And I've highlighted some of this work here, but HIV was the first case in which we actually expanded cells with the CD328 beads. Believe it or not, the first CAR trial was actually done in HIV and not cancer. And I'll talk a little bit about that in a second. The first trial that used a lentiviral vector was done in HIV. And the first study of long-term persistence of engineered T cells was also done in HIV. Now, what cancer has brought to us is that they showed us that CARs can actually work. And so that sort of then fueled the re-examination of the HIV trials, which I'll talk about in a second. And we learned a lot about how to design the vectors so that we can get one, robust expression and durable expression. And so those are some of the things I'll talk about in a second. So what are the key issues to use the CARs? So safety is obviously the most important thing, and in some sense the biggest challenge, because HIV disease is not cancer. And right now people are living very long and healthy and fruitful lives by taking their pill every day. And so for this to go forward, we need to have an exceptionally safe product to treat people with. So what you call minor toxicities in the cancer field, they're major showstoppers in HIV. So the design of the CAR will go over, how the CAR is delivered, how can we prevent the cells from getting infected, what is the dose required, and then lastly, the persistence of these cells. And so there's some really good news here. So in cancer T cells, as we talked about earlier, the off-target or the toxicity is caused by cytokine storm. Well, that's hopefully not going to be a problem in HIV, because at the time we deliver the T cells, there should be vanishing little antigen around. And then the other part is that there's on-target but off-tissue reactivity, because most therapies targeting cancer are indeed self-antigens. In HIV, we have a whole new foreign object that we can make really good reagents that are highly specific and don't cross-react. And we have some safety data. So the infusion of the CD4 CAR, while it wasn't terribly effective, it was exceptionally safe, and there was no cross-reaction. And these cells live for up over a decade in people that we could detect. So I think there's reasonable things that that infusion of HIV specific T cells will be safe, but obviously we won't know until we actually do the experiment. So the actual design of the CAR isn't terribly different than every other CAR trial, but there are some key differences I want to highlight. So in HIV, there's only one player in town. So HIV envelope is the only factor that actually gets expressed on the cell surface, and therefore it's the only thing you can target. The great news is that we have tons and tons and tons of antibodies to choose from. And you certainly can imagine many combinations of them. There's three broad classes. One are the broadly non-neutralizing antibodies. And they have been less studied, but I could almost argue that they should be better studied, because the CAR works by killing cells. It doesn't neutralize things. So I'm not sure you really gain that much by actually having a neutralizing CAR to it. But what you do need to see, you want something that's recognized without conformational changes to occur. So you want something that's always present, so that when the T cell actually sees it, it can kill it rapidly. Then the broadly, so the one thing, argument for the broadly neutralizing antibodies is that you might get a 2-for-1 deal, so that in addition to targeting the affected cells, you actually may be able to neutralize some free virus And then the last approach is actually just using the natural receptor CD4 as was done previously. There's lots of strength to this, as it's not going to be non-immunogenic. It's already been shown to be safe. And if HIV tries to escape from it, that could have some pretty bad implications for the virus. It's hard to imagine how it can escape from CD4 binding and still be infectious virus. But over the years, we've learned not to underestimate HIV. So I'm not going to say it's not possible. So just a couple, highlights of what's been done in the field. So this is a study from Ed Berger, and he had the neat idea of combining 2 recognition domains into his CAR. And I think this is something you can do much better in HIV than necessarily cancer. So what he proposed is that one would lock it, and so this dual targeting worked much better than single targeting of either. And then, something that is certainly true in HIV as well as cancer is, how do you actually tell one thing is better than another? And so the way we do it, and there's certainly other approaches, is we separate the 4s and 8s. We infect the CD4 cells, we engineer the CD8s, and then we mix in at various ratios the number 8s to see how can we control a spreading infection. And in our mind, this is a better way of doing it because control of the spread of the infection is what you really want to do in vivo. And doing like short-term cytokine assays, while they're really interesting to do, they don't really tell you how you can actually prevent the spread of the infection. So one study that we did is we compared a number of broadly neutralizing antibodies and to the CD4 CAR that we came up with. And the first thing you see is that if we used a TCR or HIV specific T cells from an individual, there the effector target ratio is actually inverted. So to really see suppression of a virus, you need 3 effectors per target. With the CARs, they work much better. And so here the effector target ratio is, it's 1 to 25. So you need very few cells, and it's hard to know, but in some of our trials, we've easily gotten 5% engraftment of the engineered T cells. So that's roughly in the range that we can get control here. And what you see is that the CD4 CAR is able to control infection better than the rest of the antibody ones. So from our point of view, that's probably a better way of going, considering the other things I mentioned previously as well. So the other thing is that might be a little different is the transmembrane domain in the cytoplasmic tail. So in cancer, you're mostly interested in what provides the best signaling. But in HIV, we're also very worried about infection, and it's known that the CD4 transmembrane domain facilitates infection. And so in the study that I'm about to show you, we swapped out the CD4 transmembrane domain for the CD8 transmembrane domain. And what you can see is, using the same CAR, just changing the transmembrane domain led to this one working much better than the CD4 transmembrane domain. And so I think that's an additional aspect in this that necessarily isn't a cancer problem. The other thing we looked at was how various costimulatory motifs affect activity. And here the CD28 domain worked better. And we're not sure whether it's because CD28 just provides more effector function, or what we've shown previously is when a T cell gets CD28 co-stimulation, it massively downregulates CCR5. And so that would make these cells less infectable. And interestingly, in this study, the addition of the TNF costimulatory members, 4-1BB, CD27, as well as the ICOS, those receptors actually enhanced infection. And what we think is going on here is there's always a tonic level of signaling that goes on when you put a CAR into a T cell. And HIV loves activated T cells. And so what we speculate here is that the tonic signaling is actually making these cells much more infectable, and that's why they're not working as well. So something that you may or may not know about, but CD8 cells actually have the ability to do CD4. It's a very transient activation, but since we are targeting these CAR cells towards HIV, any activation of CD4 is going to make those CAR cells susceptible. And so we did a study, and this sort of shows the complexity of understanding how these cells get infected. Because if you take a pure population of CD8 cells that have been transduced with a CD4 CAR, you actually don't see any infection. Because what happens is the HIV gets in, and you have all these effector cells around, and they kill the HIV, so you don't see any infection coming out. Now, as you dilute the number of effector cells you have to the point where the effectors can't control the spread of the infection, now you see these cells get infected like there's no tomorrow, and they actually are the first ones that do get infected. So while you're controlling HIV, infection is not a problem. But once you lose control, then your effector cells are the first ones that actually get infected. And it's just not related to the CD4 CAR. We saw the exact same kinetics when we used the single chain antibody constructs, probably alluding to the fact that these cells, once they get activated, they're inducing a little bit of CD4 and they get in. So it seems that to really do this well, we're going to need to have another means of protecting these cells from infection. So, something that was alluded earlier, that, CD4 help is really important for CAR therapy, and I would argue is even more important for HIV therapy. So the first cells that get infected when a person gets HIV are the HIV specific CD4 cells. And this has catastrophic effects for the immune response, because once you lose your CD4 cells, your CD8 cells get exhausted, you don't make good antibodies, and so restoration of CD4 help is probably going to be absolutely essential for an HIV cure. And so we can certainly engineer the CD4 cells, but then again we have the same problem that they're going to get infected again. And so I've highlighted a few things on this diagram here of potential ways of protecting CD4 cells from getting infected. These include using gene engineering devices such as CRISPR to knock out co-receptors, to expressing antiviral proteins that prevent the spread of the infection. TRIM5 alpha is a restriction factor that prevents entry, as well as fusion inhibitors. We recently published with Jim Hoxie a fusion inhibitor that fused a fusion innovation with CXCR4 and showed that it had great potential to prevent infection. And so then, moving on, what's the best way to deliver these CARs? And so this was a study we did in which we compared a murine retroviral vector with an HIV-based vector, but everything else was the exact same. And what you can see is that at very high effector target ratios, both work well. But as we alluded down, the murine-based vector loses control much quicker than the HIV-based control. And we think this has to do with the overall expression. So if we look at the expression of the murine-based vector, we see roughly 10-fold more expression with the HIV vector. And that may have something to do with the fact that HIV integrates into open reading frames, which are open chromatin. So it may be more simple for the transcription factor to get there to keep it going, whereas the murine goes into promoters, and those may be more silenced. And so here's a study from Thor Wagner's lab in which he had a clever idea of sort of getting a 2-for-1. So he designed an AAV approach to target the integration into CCR5. So in this way, you actually disrupt CCR5 and you get expression of your CAR. So the big question, well, and actually in all CAR fields, is how many cells do we actually need? And to date, most trials have been designed on how many CAR cells can we produce, and that's how many cells we put in. But as the field matures, we're getting to the point now where we really need to do proper dosing studies. And what I'll highlight on the left here is one of our first patients from Penn. And this is actually a great story. So this person had so much leukemia in their blood that when we did the apheresis, we really couldn't find no T cells. And so my colleague Bruce Levine had the great idea, since we used the CD328B to activate the cells, he dumped $10,000 worth of beads into the bag, shook it up, and used a magnet to actually purify the T cells. And this genius got him 1 million T cells from an apheresis bag. At that point, we were already financially committed. So we went on with the trial, and we transduced those cells, and we got 15 million T cells that we infused into the patient. And that's what you see right here. So tracking this person, this is the percent of the cells in, and you're very early, it's really at the limit of detection. But what's absolutely amazing is that over the next 20 days, that's almost a vertical line of expansion. And to this day, this person is walking around playing golf and having a very nice life. But so it really highlights that the CAR T cells are living drugs, and they will adjust their levels sort of based on antigen and need and stuff like that. For HIV, we don't have this luxury, and it means that we're going to have to, because the human body there's lots of places to go, and HIV, it's a blood disease, it goes everywhere, and so to get enough cells sort of on the front lines when you administer these latency reversal agents is not going to be easy, and so I'm suspecting we're going to need to be on the higher end of the dosing realm. And what experience has taught us is that the more cells you try to produce, the less well they engraft. And so we're fighting this battle between how many cells to put in, how long will these stay around, and then where will they go. And so unfortunately we don't have any data on this yet, but this will be a major obstacle in this type of a therapy. And then even a bigger question is, if somebody gets infected in their 20s and they go on their medicine and we put the T cells into them, and I think we'll get suppression of HIV at some point, for a durable amount of time, but how long is durable? And how long will people, will need to be monitored? And when do we say somebody's cured? Because HIV has proven remarkably, is it possible that after 20 years somebody pops up again? Sure. And so that's going to be a very tough dilemma for the patient population and the physicians to really monitor somebody after they've had, at this point it'd be a wonderful problem to have because we're not even close to that. But it is something to start thinking about in terms of, what does a durable cure really mean in HIV? So I'll leave you with sort of what our first clinical trial will be in this. And so I didn't go through all the data because this was an educational thing, but we think we've designed a really nice and effective CAR that works really well in vitro and in vivo. And we're now in the process of doing all the stuff you need to do to get a trial opened. And we designed a trial that would hopefully allow us to ask a few questions on one trial. And the way we plan to do this is to expand the cells, put them into the person while they're still on ART. And the first question is if we put in 10, 30 billion highly functional T cells and allow them to swim around for a while, if ongoing HIV infection is actually occurring, and that's something that actually maintains this latent reservoir pool, we might predict that after a month or so of the cell therapy, we might be able to see a reduction in the reservoir just by having the T cells in there, if they're able to go and kill and reduce that. Then the second arm is then use of one of these latency reversal agents to actually then induce, because the T cells are already there, they're ready to go. We add the latency reversal agent, and then we'll ask, did this actually reduce the reservoir? In full disclosure, I'm not 100% sure what that latency reversal agent is going to be at this point, because There's not one that's terribly attractive, but we're working very closely on that right now. And then the last part of the experiment, which is the exciting part, is now what if we take the person off of their therapy, how long can we get a durable control? Because if the CCR5-ZFN patients, so our hypothesis there is that we were able to make a few HIV-specific CD4 cells HIV resistant. And those CD4 cells had a dominant ability to sort of rejuvenate the immune response, and in many cases, in some patients, lead to a long-term control of virus. Well, now we're not talking about a few cells. Now we're talking about billions of cells that are going to be HIV specific and HIV resistant. And so if our hypothesis is correct, we're crossing our fingers that we'd actually be able to see a pretty sustained control of virus once the drugs are removed. And so that's sort of what our plan is. And then the other thing we'll also be looking at is, now that we restore CD4 help to the disease, can we revive the other endogenous CD8 cells that are there and make them more functional and sort of have the CAR and the endogenous response sort of work together to keep HIV down. And on that, I'm blessed with a number of awesome colleagues throughout the years. Great help from Sangamo, Carl, June, John Weary, Rick Bushman, and all the names out here. But it's been a very collaborative idea to get these ideas on the board. And on that, I'll take whatever questions.
Questions? I'll ask a question, actually. Yeah. So in the CD19 trial, you pointed out that even a few cells could expand out to a large number. Would it be better possibly to introduce antigen instead of putting in so many more cells? Because I know from a lot of trials, people found expanding cells to that level, really what you're putting in, the majority of them are going to die off.
Right, and so there have been people, there are a number of vaccine candidates that you could, postulate putting in. It just makes it more complicated to do, and whether there are good vaccines out there to keep that going, but I think that's a good idea to sort of keep the, maybe a booster vaccine every year or so to sort of keep the cells active and growing.
My other question is about the removal of the ART therapy and the timing of that. How long are you planning to wait? 4 weeks? Is that what it says, or is it?
Well, if we do all the thing, it would probably be closer to 2 months.
So I was wondering if any information on the mutation in the E and V epitope that you're going to be targeting. I think 2 years ago there was a paper coming out showing that GAG was a very immunodominant epitope for CTLs, but if the patient didn't receive HART therapy soon enough, then in vivo the HIV virus has lots of mutations in GAG, and there's lots of immune escape variants. I was wondering if you had observed similar things in M, for it's a very stable protein.
We don't know yet because we actually haven't done the trial yet. In vitro studies, they don't really mimic escape terribly well. But that's, and to be honest with you, that's why we sort of favor the CD4 approach, because we think it's going to be hard for HIV to escape. Certainly, any antibody response or most antibody responses, HIV is pretty good at escaping from. But yeah, we don't really know whether how much escape we're going to see in this.
Thank you. So I have a question regarding whether when the free virus binds your CAR, whether CCR5 is there or not. If the CAR is activated, it probably internalizes in the cell. Will that bring in the virus in the cell and become infected? That's a mechanism possible?
It's possible, although the amount of free virus out there is probably pretty minimal when we do this stuff. And so it's generally thought in vivo that most spread happens from cell to cell. Which still brings up the same problem, actually.
Okay.
But yeah, I think that's the danger of this, is that by targeting HIV, you're actually bringing it closer to its prey. And so that's why we think you really need to have a very strong and robust way of preventing those cells from getting infected.
Thank you.
Yes.
Good question. So, continuing your logic with the cell-cell transmission as a dominant way, how is your approach coping with that? And the other question, is it my understanding
I mean, that's sort of the same, some of these issues also have cancer, right? I'll repeat the, so the first one, oh, help me. The cell-to-cell transmission. So, will the CAR T cell prevent cell-to-cell transmission? So like all T cells, I mean, as long as the envelope's expressed on the surface, they'll kill and it will prevent the cell. It's a race, right? So what you need to do is kill the cell before it makes spreads. And that's what we model in the in vitro studies is how quickly can we kill the cell before it spreads. But if you get it early enough, you can block the cell-to-cell transmission. The second question is GP120 is often shed, and and that could bind to other cells, and that could lead those cells to getting killed by the CAR. And that absolutely is a possibility. And I think the same issues happen often in many of the tumor antigens that we target, that many of these antigens are also spread, and I think that's just life in wartime. I think there's going to be some innocent bystanders in any type of therapy that you do in this.
So it was interesting that you mentioned trying to see whether patients on the CARs will be really effective when patients are on ART. How would you be able to detect that? And if you don't see it, how would you distinguish whether it's because there's not replication or whether your CARs can't get at HIV, for instance, if it's replicating in like a follicular reservoir or something?
Yeah, you bring up a good point. I mean, so it's one of those experiments that if you see something, you believe it. Mm-hmm. But if you don't see something, you probably don't learn all that much. And so there's very sensitive ways of actually measuring the reservoir now. And what we're planning to do is actually sequence. And there's a ratio of viruses that can make real viruses, and then there's a lot of viruses that are completely defective. Mm-hmm. And what we would try to do is what's the ratio before treatment and after treatment. So what we predict is that the CAR therapy is going to target the replication competent, but it's really not going to target the replication incompetent because that'll never get expressed on the surface. So by looking at that ratio, we'd hopefully get something there. But if we don't see a change, I'm not sure what it tells us, to be honest with you.
Thank you.
Great, thanks.
So that's the last speaker. Thank you very much for everybody who came.